So my name is Alfred. I'm the Director of Research at Etcembly. And Etcembly is an AI tech bio based in Oxfordshire. And today I will talk to you about the technology behind our AI engine, known Emily, and what Emily has achieved so far. So Etcembly was established in 2020, literally just at the height of COVID when we got our first funding. And the aim of our founders at the time was to combine their experience in drug development and computational biology to create an AI platform that not only we can use, but that others in the drug development field can also exploit and to harness for their own drug development. And 6 years on from that, this is exactly what we have developed. So on the middle panel there on top, in this last 6 years, we have built and improved our own AI engine known as Emily. And Emily has been trained using millions and millions of sequences of immune repertoire sequences, antibodies and TCRs, either from healthy donors or from cancer patients itself. And the idea is that we want Emily to recognize what does a natural immune complex look like, how they bind, and what do those structures differ from one another. And in 2023, we came out of stealth mode when we became one of the first companies to actually use Emily as a generative AI platform to design and also engineer a TCR-based biologics all the way to ultra-high affinity. And we managed to essentially cut down the lead discovery and engineering time from the conventional 2.5 years to just about 11 months, so representing a cut of up to 60%. And through a partnership project as well, we have recently been told that one of the molecule that Emily has generated has now been accepted for a clinical trial. And trials are bound to be started in the UK and also in Spain later this year. So we are very proud, but also very excited to have contributed to that project. And over the last 6 years, we have filed 5 different patents for the different assets that we have developed over the last few years. So on the right panel are the different partners that we have worked with, during this duration. They all range from infrastructure AI up to basic AI ML solutions. So yeah, we'll talk about that a bit more. So what we feel proud about Emily is that Emily has been validated not just internally by us, but also through some partner projects. So on the right-hand side here is our own internal portfolio development, where we have developed a bispecific T-cell engager against the commonly known cancer target PRAME. And what we show with the graph here on the right-hand side, on the left-hand side, sorry, on the pink, is that the bispecific T-cell engager that we have developed shows significantly better potency and sensitivity in killing a patient-derived organoid sample compared to a clinical competitor benchmark that is more commonly known. And if you look at the graph on the yellow, just on the other side, you can see that the competitor drug could only work if we actually add more target to it, indicating that the EC50 and the sensitivity of the drug that was designed by Emily is a lot better than what is already out there. And on the right, on the other hand, is a first-in-class cell therapy modality that we have developed with another Norwegian company called Zelluna. So Zelluna approached us because they have a lead molecule that is good but not potent enough. So what we have done is to increase that potency of their drug quite significantly. As you can see from the dose response curve there, so the dotted blue represents the parental molecule that was presented to us. We were able to generate 2 variants which has an improvement in EC50 by more than 100-fold through only a single round of affinity maturation. And this translated into a much better increased efficacy or cytotoxicity when this modality was used to kill its own cancer target cells here. And this was the lead molecule that was shown to be safe and tolerable and received the approval for a first-in-human trial in Europe last year. So I mentioned earlier that one of the ways we train Emily is to give it millions and millions of sequences and structure of immune complexes, particularly with antibodies and TCRs. And here I've separated antibodies and TCRs, antibodies in green and TCR in yellow. And why we do this is because the first step in our drug discovery pathway is that we want to make sure Emily is able to model the immune co-complexes correctly. And what I really want to highlight here is that the figures that are in the middle is to show how the model that are generated by Emily is able to superimpose on the experimentally determined crystal structure with an RMSD of less than 3, indicating there is no difference at all between our modeling and the actual crystal structure. We expanded this analysis, beyond our own in-house capability to more than other 40 complexes that we can retrieve from public domains. And here you can see for both antibody antigen complexes and TCR complexes, we were able to model this with a median RMSD between 2 to 3, indicating that how accurately Emily is able to do this. One question we get asked a lot is that what contributes to this accuracy? And this is also down to the way we select those models after we make them. So we have developed our own proprietary tool or algorithm, which we have called Doriad. And what Doriad does is that it looks at the different conformation of the poses of the co-complex that is being developed. And Doriad then screens them one by one to understand which of these co-complexes actually represents the native configuration and which of them actually represents the biological reality. And by doing so, we analyze the different dynamics and poses of binding to allow the efficient ranking of the different models that we have developed here. And there when we compare the performance of Doriad with other conventional tools and open source tools out there, such as DeepRank and GNN-Dove, we managed to show that we were successful in more than 50% of the time as well. So this paper is currently under review under Nature Structural Biology, but it is retrievable on bioRxiv at the moment. So one question that we get asked a lot as well is that, there is a lot of protein modeling out there, made famous first by AlphaFold, but more and more generations of protein modeling tools have come up, like Chai and Boltz as well. And we get asked a lot, how does Emily compare to all these very widely used and open source tools? And here we show that comparison. So the blue and the yellow represents the different complexes, so complex 1 and 2, and the different labels there indicates, whether they were modeled using our software or open available software. So we look at the RMSD and the DocQ scores. So if there is accuracy in the modeling, this is represented by a small RMSD, but a high DocQ score, which is exactly where Emily sits relative to the other open source like Boltz and Chai too. And on the right here, I wanted to show exactly how those models sit. Blue represents the experimentally determined crystal structure. Green is our model, and brown and pink represents Boltz, Chai and Boltz respectively. And you can see that out of the three softwares here, only Emily is able to accurately represent the variable domain and the variable loops of the CDRs in this instance. And both Chai and Boltz too actually failed to actually model one of that CDR completely, and it's out of range compared to the experimentally determined crystal structure. So why are we obsessed with accuracy of modeling? Because if you can model accurately, you can actually design drugs that bind well to its target, and you prevent the issue with cross-reactivity. So on the figure on the left here, this is what we wanted to show. So we retrieved more than 500,000 VHH antibody sequences from these proceedings in 2023. So from this data set here, they have already determined which of these antibodies binds their target antigen, which is IL-6, and which do not. And what we did was to take this collection of antibodies, do a completely blind experimental data of clustering using our system, and see, can we actually identify the binders from the non-binders? And this is exactly what we have managed to do. You can see that the vast majority of the cluster plots here are blue, indicating that most of them do not bind, which is what you expect from an antibody campaign. But the red represents the binders. And what we were surprised as well is that even with extremely small clusters indicated by A, B, and C, Emily is able to actually accurately determine which of these really small clusters are very much enhanced or enriched actually for the presence of an actual binder, meaning that if we go into those, there's a higher chance actually of us selecting the correct antibody that binds to your target antigen. We then applied the same principle to do affinity maturation, shown on the right here. So this is after just one engineering round for a partner project. And here, what we wanted to show is that when we select a minimal number of variants, which is just slightly under 100 here, in both cases that we are trying to affinity mature, the large proportion of the predicted variants of the 97 actually retain binding to its target antigen. And between 25 to 80% of those antibody that we have predicted were actually of higher affinity than the wild type, which is what you want. And we are looking at a range of a 3 to 5 increase in affinity maturation fold here. So what this means is that after this one engineering round, we then have an existing lead molecule that we can then engineer a little bit further on to get to the final number that researchers are looking for in a drug. So we put all this sort of clustering analysis and modeling to a test, and last year we completed a case study with DGS antibody, which is a subsidiary of AbbVie in Oxford, sorry. So what they came to us with, so AbbVie has done the immunization of the mouse with a target antigen, and they came to us with their collection of B cells. And the idea here is that, if we sequence those B cells, can we accurately identify which of these binders are actually good binders to their target. And this is what we did. So we took the 70,000 antibody sequences from them, put it through our pipeway, do the sequencing clustering as you see before. And what is interesting is that when we do those clusters, we were able to actually identify more than 400 clusters which sequence variation, but also with different modes of binding to their target antigen of interest. So we are just showing here 3 out of that many of them. You can see clusters A, B, and C. Each shows minor but significant variation in their mode of binding. And from here, we were able to do a ranking and then select out of 70,000, 180 variants that we returned to them for validation. And out of these 180, more than 30 were actually binders to the target antigen. And out of these 32 of them actually had 5 times the signal compared to a benchmark antibody that they've been using. So this represents a significant savings both in time and cost. That filtering from 70,000 mouse antibody sequences all the way just down to the 2 final ones. I'm going to finish my penultimate one. So by training Emily on a lot of antibody sequences, in addition to doing target binding and affinity maturation, we also look at how this affects manufacturability of an antibody. So on the left here is a partner project we did with Vector Labs. So the pink represents the parental antibody that was given to us, and which, they had a lot of issues in the lab, not just with yield, but also with aggregation issues and things like that. So using Emily, we were able to generate 12 variants for them, return it to them, and more than 80% of the Emily engineered variants were actually expressing significantly better than the parental antibody. And so this experiment was repeated twice by them, not us, and they know that therefore the result is reproducible. And what they have found is that in addition to improve in expression, the aggregation issue that they have seen previously has also completely disappeared, indicating that we didn't just increase the expression, we also improved the solubility property of this antibody. And on the right is one of the interesting ones. So we have been looking at protein sequences all this while. We decided to now look into nucleic acid sequences. And when Twist came to us for this project, one of the instructions was that this antibody cannot have any mutation. It is almost the final molecule, and therefore we can only play with the codons and not with the protein sequence. So we developed our own codon optimization sequences, not codon optimization tool, sorry. And from the 3 parental antibody that they gave us, just by virtue of changing the codon, touching the nucleic acid without changing any protein sequence at all, we were able to have a success rate of more than 96%, where almost every prediction that we have made here has increased the expression over the wild-type molecule itself. So this is something that we are discussing with a lot of CDMO at the moment, partly because when molecules will reach the CDMO stage, it is the lead molecule that can no longer be mutated. Hence, looking at codon is the best way to go forward. So I'm going to skip this in the interest of time. So I hope in this slide here, in this presentation, I have given you a flavor of what Emily can do. And using Emily, like I said, we have actually launched our own innovative T-cell engager programs. And to date, we have developed a preclinical asset which we believe is a best-in-class tri-specific candidate. And what fuels Emily, in our ability to sequence millions and millions of those sequences is that in-house, we have also developed a very ultra-high throughput microfluidic single-cell platform. We have begun offering this platform to other people who might be interested in also sequencing T cells and B cells. So, if this is of interest, please do get in touch. And with this, I end my talk. Thank you.